Water is 800 times denser than air, generating profound resistance for any organism moving through it. To maintain continuous momentum in this environment, pelagic fish rely on a highly specialized internal architecture. Looking at this anatomical cross-section, the propulsion system is clearly divided. This is a strictly partitioned bimodal locomotor engine. The dark red tissue concentrated along the lateral line is the oxidative muscle. It is a slow-twitch engine, packed with mitochondria and fueled by a continuous supply of oxygen to power sustained cruising. Surrounding it is a massive block of pale white glycolytic muscle. This is the fast-twitch engine, capable of generating massive explosive force through anaerobic pathways for brief, high-velocity movements. Traditionally, biologists measured swimming performance as a monolithic trait. They averaged total oxygen consumption against speed, effectively blending the distinct outputs of these two muscle types into a single, less precise number. Modern respirometry and deep RNA sequencing have provided a more granular view. We can now observe how these tissues operate under separate metabolic rules and divergent genetic timelines. Mapping the precise energetic cost to these specific tissues demonstrates that high sustained reforming our understanding of how aquatic locomotion actually scales. This plot charts red muscle's baseline aerobic efficiency. As speed increases, oxygen consumption rises steadily, but this has a fixed ceiling. Before accumulating anaerobic products, the fish reaches its maximum sustained swimming speed, or EUSIS, the edge of its aerobic capacity. When velocity exceeds EUSIS, the red muscle can no longer meet the demand alone. The brain recruits the white, fast-twitch muscle to bridge the gap. The tail lashes, generating a sudden surge of kinetic power. The fish has transitioned into unsteady, burst-assisted swimming. These bursts provide immediate speed, but they break the organism's metabolic equilibrium. The white muscle burns through internal reserves that cannot be replenished in real time. To quantify this anaerobic effort, researchers measure excess post-exercise oxygen consumption, or EPOC. It's a metabolic debt repaid with elevated oxygen intake once swimming stops. By isolating this recovery in gildhead sea bream, researchers correlated debt size with individual bursts. The data shows a rigid penalty for this unsteady motion. Every physical burst executed by the fish incurs a linear anaerobic debt of exactly 0.53 milligrams of oxygen per kilogram of body mass. While the white muscle engine is expensive, the red muscle engine exhibits a surprising degree of efficiency during long-distance cruising. This scatterplot measures sustained travel, maximum sustained speed against minimum cost of transport. In 2012, a morphological model proposed a trade-off. Organisms optimized for speed would be less efficient. Graphically, this forms an upward-sloping line, linking increased speed to higher costs. But empirical data from S. aurata and P. reticulata shows the opposite. The theoretical trade-off doesn't exist individually. Plotting the actual physical performance reveals a downward trajectory. Individuals with the highest recorded speeds actually achieve the lowest costs of transport. This negative correlation indicates that the physiological traits for speed and economy are synergistic. In axial swimmers, the most powerful aerobic engines are also the most efficient. Evolution does not require a compromise here. High-performing individuals maximize their uses while simultaneously minimizing their energy expenditure per meter traveled. How does the red muscle expand this efficiency? To find out, researchers subjected rainbow trout to a constant 4-week swimming regimen. This chart tracks the initial transcriptomic response. During the first week of exercise, shown in these gray columns, the red muscle senses the physical stress and initiates a massive genetic surge. mRNA expression for regulators like AMPK and PPAR-alpha increases by up to 36-fold. The tissue is signaling for a complete metabolic overhaul. However, there's a disconnect between genetic signaling and the muscle's physical capacity. Despite the surge in mRNA signaling, concentrations of citrate synthase and hexokinase, markers of metabolism, remain entirely flat through weeks 1 and 2. By the 4th week of continuous swimming, the transcriptomic signals have subsided. mRNA levels for those regulators have returned to baseline. It is only now, at week 4, that we see the physical result. The concentrations of citrate synthase and hexokinase finally spike. There is a profound temporal lag in endurance adaptation. It takes a full month for the genetic instructions sent in week 1 to manifest as doubled enzymatic capacity and mitochondrial biogenesis. Under the same conditions, the white muscle shows no such remodeling. Its genetic profile remains largely unchanged, reinforcing its role as a fixed anaerobic reserve. This remodeling is strictly partitioned to the red muscle. The tissue requires weeks of sustained stress to translate rapid molecular signals into a higher-performing physical state. This lab-measured lag between genetic signaling and physical change provides the framework for one of nature's most grueling migrations. Wild Pacific salmon must travel thousands of kilometers upriver to reach their spawning grounds, a feat powered entirely by the bimodal engine. The transcriptomic surge observed during the first week of the trout trial aligns with the molecular sequence identified in wild salmon during their ocean-to-river transition. In both cases, the tissue initiates remodeling before the physical demands reach their peak. As they move inland, the salmon reserve their white muscle for high-cost anaerobic events, like leaping over waterfalls. Because they have stopped feeding, this white muscle is eventually cannibalized. The body breaks it down for protein oxidation as lipid stores are exhausted. Meanwhile, the red muscle handles the thousands of kilometers of sustained cruising. Its extreme plasticity, utilizing rapid genetic signaling to drive delayed structural changes, shows that the red muscle engine is built to relentlessly optimize the cost of transport to ensure the survival of the species.